Good afternoon, everyone. Today, I'm going to present the result of a simulation study designed to explore genotype-by-environment interactions in dairy cow feed efficiency. This work has been done thanks to a collaboration between two worlds, the worlds of genetics with Peter Eimer and Vincent Ducrot, and the world of nutrition with Nick Friggans and myself. Today, breeding for more efficient cows is a key challenge for the dairy sector. We need cows that make the best use of limiting feed resource, and we need cows that produce few wastes such as methane emissions. The selection strategies to achieve this breeding objective should be sustainable, and in terms of research, this has two major implications. First, we have to ensure that improvements of efficiency in the short term are not leading to detrimental effects in the long term and are not impairing animal resilience. Then we also have to evaluate if selection strategy for feed efficiency will still be appropriate in face of changing environments. In other words, the question is not only to select for high merit cows for feed efficiency, it is also to evaluate if those cows will still be resilient and able to accommodate different environments. These two facets of the problem leads us to the objective of our study, evaluating genotype by environment interaction for feed efficiency in the short term and in the long term. Before looking at the results, I will give you a few insights in our method. I'm sure in this session, we all know that when working on efficiency, the most important aspect is how to deal with the complexity of these traits. First, it's a trait that is very difficult to measure in the field. That's why we choose a simulation approach, which is a complementary method to other data-driven approaches. Efficiency is also very complex because it's a composite trait made of subtraits such as body reserves or fertility. And all these traits are changing with time depending on the animal life stages. Finally, they may also have some trade-offs among those traits. To catch these complex features of efficiency, we developed a model that is based on physiological mechanisms and that will simulate lifetime trajectory based on the resource allocation framework. Let's now look at the biological building block that we use to generate phenotypic traits behind efficiency and further evaluate genotype by environment interaction. The individual dairy cow model is dynamic and simulates the dairy cow lifetime from birth to culling or death. You can see an illustration of this dynamic feature on the graphs above the cow. Then we need to have biologically consistent links between the different traits behind efficiency. We also need a link with the environment. For these reasons, we use nutritional principles that allow us to connect the energy available from the environment to quantitative traits. These principles are static equations. They are just reflecting how energy is used. To introduce dynamics, we consider two key drivers in the model. The first driver is energy acquisition. It reflects the innate capacity of the cow to acquire resource. The second driver is allocation, which reflects how the energy acquired from the environment is distributed between biological functions such as growth or lactation. Allocation reflects the inherited dynamics of priority of the animal depending on its life stages. We choose to use the allocation framework as the conceptual basis of the model because it is a way to integrate trade-offs directly in the model structure. With these two drivers and with an environment providing feed resource, the model simulates the lifetime trajectory of the cow and the different traits. An important aspect to bear in mind with a model based on mechanism is that traits are built during the simulation. So for instance, the number of lactation and the longevity of the simulated cows are the result of a simulation. Now that we have a building block to generate traits and study efficiency, we need to move on at population level to introduce variability, which is essential to study GBAE. We use a straightforward approach with individual-based modeling. Each single cow corresponds to an individual model, and the population is created by multiplying individual models. We then need an environment to have the population eating and reproducing. This environment corresponds to feed resource dynamics defined by dry matter offer and energy content of dry matter, and also to reproductive and culling management rules. Finally, to obtain a diversity of lifetime trajectory and variability of traits, we need to introduce differences among animals. We choose to do so with the parameters behind acquisition and allocation. We consider 4 of them as genetic scaling parameters reflecting different genotypes. By drawing randomly those parameters in a distribution, we are able to simulate a diversity of lifetime trajectories and variability in efficiency metrics. At this level, we only have created phenotypic variability, which is of course a problem to study G by E. Let's look at how we introduce genetic variation. We developed a module to create a virtual population of sires that have breeding values for the 4 genetic scaling parameters. Based on some assumption on heritability and genetic correlation, we created those breeding values, and together with a pedigree structure, these breeding values are used to assign the value for each cow in the population. So now, if we simulate the population of cows in an environment, we get phenotypic values for the different traits, but we can also compute genetic parameters. If we run the same population in another environment, we also obtain phenotypes and genetic parameters. By comparing the genetic parameters, we can explore genotype by environment interaction. Now that you have the big picture of our method for exploring G by E, let's look at how we define the scenarios for this study. We consider the population of 20,000 cows from 200 sires. We choose to study grass-based farming systems as they are more likely to be challenged by resource availability in the future. Calving is seasonal, and cows are culled if they calve after the desired calving period. We define 4 scenarios of resource availability represented on the left graph on the slide. The first scenario is not on the graph because it's a non-limiting scenarios, and you can see that the other one reflects seasonal variation in feed offer, and they differ in terms of level of feed availability. I will not detail the scenario, but on the right side of the slide, you can see the constraints imposed by the different scenarios, ranging from 0, meaning that all animals can express their acquisition potential, to an average of 50% restriction compared to the potential acquisition. Let's now look at the results. After simulation, we ended up with a population of 80,000 virtual cows, corresponding to 20,000 cows in 4 environments. Based on the simulated traits, we computed 2 different metrics of efficiency, always as the ratio between the energy invested into milk over the energy acquired from the environment. The first metric is a short-term metric at the scale of the second lactation. The second metric is the long-term efficiency metric, computed at the scale of the whole lifetime. We can see that the environment did not affect efficiency at the second lactation scale, while there was a strong effect of the environment for lifetime efficiency. This effect is mainly due to a decrease in the total number of lactation and longevity when the environment became more and more constraining. Regarding genetic parameters we obtained, I only present the results for the 2 most contrasting environments. We estimated heritability and genetic correlation for the 2 efficiency metrics, short-term and long-term, but also for other traits related to production and reproduction. I don't have time to comment everything, but we can see that the correlation between lactation efficiency and lifetime efficiency decreased when the environment became more limited. We can also see that within a good environment, body reserves At second calving and lifetime efficiency were not very correlated, and this correlation increased when the environment became more limited. To go further in understanding genotype by environment interaction, we looked at the strategy behind the best cows. We compared the strategies of the best cows for efficiency in the short term with a high level of resource, with the best cows for long-term efficiency with a low level of resource. On the graph, you can see the distribution of the two acquisition parameters for the best cows compared to the whole population. The only difference was for the best cows for life efficiency. They had a lower basal acquisition parameter, which is related in the biological model to a smaller mature size. If we now look at allocation parameters behind the best cows, we can see that these parameters are much more discriminant. For allocation to growth, the best cows for long-term efficiency with a low level of resource, the value of the parameter is lower for those cows, which means that they are allocating more to their body reserves. If we look at lactation allocation, we can see that the best cows for short-term efficiency with a high level of resource are allocating much more to lactation than other cows. To conclude on this study, our simulation have shown that changing feed resource availability impact genetic correlation and also impact the best biological strategy to achieve feed efficiency. Further, we've seen that targeting short-term efficiency may not always correspond to long-term efficiency, which highlights the importance of having balanced breeding goal. Finally, as a broader conclusion to this work, I hope you're convinced that simulation based on biological framework can be useful to better understand feed efficiency, and also that collaboration among disciplines is very fruitful, especially when addressing such complex issues. Thank you for your attention.